When looking at the cell under the light microscope, the very first feature that becomes evident is that the cell is surrounded by a membrane that defines the boundaries of the cell. That membrane is the one that we refer to as the plasma membrane. Now, the second feature that comes to mind is the presence of a nucleus when you look at eukaryotic cells. And the nucleus defines a set of other organella that are bound to the same type of membranes inside the cell. And then the question comes to mind. First of all, what is the composition of those membranes that define the eukaryotic cell? And second of all, if there are proteins that are present in those membranes, how do proteins actually make it there? And that second question is the main theme of this new chapter in our class, in our course. So now we are going to be dealing with protein sorting and transport. And the big question that we will be trying to answer throughout this section of the class is how are proteins targeted to different organella in the cell? And what happens during the process of targeting proteins to those different compartments? The traffic of proteins within the cells to the different organella was initially defined by the so-called pulse chase experiment. There is a movie that deals specifically with this type of experiment. Please watch the movie. Now, the ultimate conclusion that was derived from this particular experiment was that proteins that were destined to the outside of the cell started their traffic within the cell at the level of the endoplasmic reticulum. And after spending a few minutes in the endoplasmic reticulum, proteins will then move to the Golgi apparatus. And then from there, proteins will move on to the so-called secretory vesicles. And from there, proteins will then be secreted out of the cell. So these define a very specific traffic pathway within the cell, which was referred to as the secretory pathway. Now, this whole pathway is summarized in this movie, so pay attention now. What you're looking at right now is a red strand that represents the RNA, the messenger RNA. For proteins that are going to be destined for the secretory pathway, that messenger RNA will be bound to ribosomes that will be associated to the membranes of the ROV-ER. Now, that messenger RNA has been translated into a protein, which is represented by the blue strands that you see there. And one of the blue strands is actually going to places in the endoplasmic reticulum where buds or vesicles are formed. And those vesicles then traffic to the cis Golgi. The vesicle fuses with the cis Golgi and then it matures into the median Golgi. Some of the vesicles from the median Golgi go back into the cis Golgi, and that is what we refer to as retrograde transport, and that also happens at the trans Golgi network. Now, some of the vesicles generated at the trans Golgi will actually move forward and become secretory vesicles that have the ability to then bind to the plasma membrane and deliver their content to the outside of the cell. Now, some of the proteins from the outside of the cell have the ability to go back into the cell by endocytosis, forming endocytic vesicles that then fuse with the lysosome. Now, the components that are present in the lysosome are directly trafficked from the trans-Golgi network. The question of how are proteins destined to go to one particular location within the cell or another is something that is defined early on during the life of a protein. Generally speaking, proteins that are destined to remain in the cytosol or in the nucleoplasm are synthesized within free ribosomes in the cytosol. And remember, this is an important point. Protein synthesis in eukaryotic cells takes place in the cytoplasm. So if proteins are synthesized there in ribosomes that are free floating in the cytoplasm, those proteins are destined to pretty much either stay in the cytoplasm or to be later on trafficked to the nucleus. Now, there are a few additional compartments that those proteins can actually be targeted to, and one of them is the mitochondria. The other two compartments, the chloroplasts and peroxisomes, are alternative pathways that can be followed by some of these proteins that are synthesized in free-floating ribosomes in the cytosol. However, if the protein is to be targeted to the main membrane-bound organelles within the cell, that is, to the endoplasmic reticulum, or to the Golgi apparatus, or to secretory vesicles, or to lysosomes, or even to the plasma membrane, or just to be secreted outside the cell, proteins need to be synthesized in ribosomes that are bound to the membranes of the endoplasmic reticulum. So the very first stage in targeting proteins to the membrane-bound organelles is defined at the moment of protein synthesis. And one important thing to keep in mind about that decision-making process is that the ribosomes that are going to be involved in the synthesis of proteins that will remain in the cytosol, as well as proteins that will be targeted to different organella within the cell, are exactly the same. There are no differences between those ribosomes. So then, how does a ribosome know whether to continue translation in the cytoplasm or to move and become attached to the surface of the endoplasmic reticulum? In other words, how do you define the final destination of a protein, taking into consideration the fact that this is going to be a decision that is made very early on in the life of the protein, that is, during the actual process of protein synthesis? And the answer comes down in the same form as the other cues that indicate where proteins must go within the cell, that is, in the form of protein signals. In this particular case, the protein signal is called an ER signal sequence, or ERSS, or simply a signal sequence, or SS. Now, those signal sequences are usually located at the amino terminus of the protein, and those signals are recognized by a complex of RNA and protein known as the SRP, that is the signal recognition particle. So, what is a signal sequence?
Banana.
It is a sequence of anywhere between 12 and 15 amino acids that are very highly hydrophobic in nature. So any sequence of 12 to 15 hydrophobic amino acids can constitute a signal sequence. And this sequence is sufficient for recognition by the signal recognition particle, which is shown here. This is the main structure of the signal recognition particle. As you can see, it has an RNA component shown here, and it has 6 different polypeptides, 6 different proteins that are associated to that RNA. And this signal recognition particle is the one that then recognizes this hydrophobic amino acid sequence in the protein that needs to be targeted
to the secretory pathway.
This means that proteins that are destined to remain in the cytosol do not contain a signal sequence, whereas those proteins that must be targeted to be synthesized in association with the membrane of the endoplasmic reticulum do contain a signal sequence, shown in this slide in red. So how does this actually take place? The whole process of recognition by the signal recognition particle is exemplified in this movie. So pay attention to this process now.
Protein synthesis for both proteins that will remain in the cytosol as well as proteins that will become associated to the ER starts in the same place, in the cytoplasm. However, when the peptide that is being synthesized contains a signal sequence, that sequence is recognized by the signal recognition particle, and that particle then brings the ribosome in close proximity to the ER membrane, specifically to the signal recognition particle receptor. This receptor then becomes associated to an aqueous channel known as the translocon. And the proximity of the ribosome to the translocon opens up the aqueous channel, therefore allowing the elongating peptide to become inserted into that aqueous channel. Now, once the signal sequence becomes exposed to the inside of the ER, it gets cleaved. by the signal peptidase. The peptide continues to be synthesized and continues to translocate through the aqueous channel of the translocon, and then the protein is fully released within the endoplasmic reticulum, within the lumen of the endoplasmic reticulum. The folded protein that results can then continue its path through the secretory pathway. So the process that we have just talked about is pretty much summarized in this particular slide, where you can clearly see how the ribosome gets in close proximity to the ER membrane thanks to the binding of the signal recognition particle to the signal recognition particle receptor, which then becomes associated to the translocon. Okay, so this is the way proteins that are destined to be secreted out of the cell are synthesized. So then the question comes in terms of those other proteins that are destined to remain associated to the membranes itself. So what about those proteins? The proteins that will be incorporated into the membrane and will become integral membrane proteins of the endoplasmic reticulum, the Golgi, the lysosomes, or the plasma membrane. For these proteins, the process will be slightly different. In this particular case, you might or may not have an endoplasmic reticulum signal sequence. Now, if you have your signal sequence, it will target the protein to the ER membrane the same way that the ER signal sequences function for proteins that will be secreted. But then, after the ER signal sequence, there will be another sequence, also hydrophobic in nature, that will act as a stop transfer sequence. This other sequence will actually be made of a longer stretch of hydrophobic amino acids. That is, while 12 to 15 amino acids is good enough to actually form a signal sequence, in order to have a sequence that will allow membrane insertion of the protein, the number of hydrophobic amino acids has to be longer. And in this particular case, it has to be longer than 15 amino acids, longer than 15 hydrophobic amino acids. And those sequences will then constitute transmembrane domains within the proteins that they are incorporated into. Now, this is a good representation of what a transmembrane domain looks like. Most transmembrane domains adopt the structure of an alpha helix, as shown here. Now, alpha helices are particularly good at crossing membranes because the natural tendency of amino acids is to form hydrogen bonds with its environment. And in a transmembrane domain, that's not possible. So that tendency to form hydrogen bonds is neutralized from one amino acid to the other in an alpha helix. When you look at these alpha helices in cross-section, this is actually what you see. You see a structure that is very narrow and in which the only things that are sticking out of the alpha helix are hydrophobic side chains that correspond to the amino acids that form the alpha helix. Now, these transmembrane domains allow proteins to be permanently anchored to membranes. And since they are permanently anchored to membranes, they will have specific orientations in those membranes. They may either have the N-terminus oriented toward the cytosol, or they may have the N-terminus of the protein oriented toward the lumen of the ER. There are proteins that not only have one transmembrane region, but actually have several of those transmembrane regions, and these proteins are actually quite important for cell physiology. One concept that is very important is the fact that the topology, that is the orientation of these integral membrane proteins in the membrane, will be maintained throughout the secretory pathway. That is, if the head, the globular head of this protein is oriented toward the cytosol, during the early stages of the secretory pathway, that is when it initially gets inserted into the membrane of the ER, that same topology will be maintained in the vesicles that will take the protein from the ER to the Golgi, and then across the trans-Golgi network, and then all the way to the secretory vesicles that will then allow the protein to reach the plasma membrane. And this is a very essential concept that is very important for you to keep in mind. The topology of integral membrane proteins is maintained throughout the whole secretory pathway. At the level of the process itself, the insertion of transmembrane domains into biological bilayers involves the translocon. And to understand how this actually happens, it's important to take a different look at the translocon. That is, to look at it from either the top or the bottom of the translocon. And this is the way it actually looks like. It looks like a circle, but it's a circle that is made by 3 different proteins, at least 3 different protein complexes. And the fact that there are different protein complexes allows then these transmembrane domains to be pushed through the space in between those protein complexes, so that now the protein can be pushed into the membrane, into the lipid bilayer. So the translocon plays a key role in allowing the insertion of transmembrane domains into those lipid bilayers. The final orientation or topology of these integral membrane proteins can be defined during the actual synthesis of the protein by the initial orientation that is adopted by the first transmembrane domain that is inserted into the membrane. And as you can see in this specific figure, it is possible to insert the proteins with different initial orientations in that membrane. Proteins that contain multiple transmembrane domains will go through the process of having those transmembrane domains being pushed into a lipid bilayer several times. And the final concept that is important for you to understand is the fact that those transmembrane domains will always be made of amino acids that are very hydrophobic. And based on the hydrophobicity and the hydrophilicity of amino acids in the linear sequence of a protein, it becomes then possible to predict which regions within a protein will be likely to be transmembrane domains. In this particular example, we see a very short segment that is not long enough to be able to traverse the membrane, and therefore is likely to be a signal sequence. A sequence that is hydrophobic enough to be able to be recognized by the signal recognition particle, but that is not long enough to be able to span the membrane. And then that's followed by other sequences that are very hydrophilic, as shown here. And then there is a segment that is very hydrophobic, and it's long enough. As you can see here, this is a measure of the amino acid number. So this sequence here is about 20 amino acids long, which is long enough to be able to span the membrane. That is, it's able to function as an actual transmembrane domain for this protein. So this region shown here then is likely to be a transmembrane domain. So just by looking at the linear sequence of a protein, you're able to predict which regions within that protein are likely to be regions that will allow the protein to become anchored to a membrane. Now, the one thing that we are not able to do is to predict the actual topology of the protein in the membrane. That is, at this point, we don't have enough tools to be able to tell whether the end-terminus of the protein will be facing the cytosol or whether it will be facing the lumen of the ER. And therefore, to determine these, you actually need to do actual experiments. Well, with this, we finish another movie. Thank you for watching. Banana.